All right, so like I said before, I'm really excited to be with everyone, and today I'm going to be talking about bacterial communities and their impact. So I'm going to start off by going over the outline of my talk. This is not okay. So the outline of my talk. Also turn on my Lisa pointer just so it's easier for everybody to see. So first, I'm going to be talking about what bacterial communities are. So what are they? And then I'm going to talk about why we should care about bacterial communities. And then I'm going to talk about the impacts of bacterial communities. And then I'm going to go on to talk about how bacterial communities can be exploited for public health benefits. And then I'm going to conclude, and then I'm going to take any questions that many people might have. So starting off with, what are bacterial communities? Okay, so when I was growing up, and even up until when I was an undergrad in the University of Ibadan, I think that my idea of bacteria was that bacteria were, they were single cellular organisms that exist in nature. They exist as unicellular organisms, so single cellular, and they are microscopic in nature, meaning that they can only be visualized by the use of a microscope. And my idea of bacteria at the time was that they were very vulnerable. And so because they exist individually and they're really small cells, if we trample on them, they're going to die. But research over the years have actually shown that bacteria they predominantly actually exist as a community, and they do not, and that many more often than not, when you see them existing as individual cells, they're actually in their transition state where they're going to form another community somewhere else. And we're going to talk about that further down as we go along in this presentation. So the general assumption, like I said before, is that bacteria exist as unicellular organisms that are very vulnerable, and they're so susceptible to harsh weather conditions. They're susceptible to being trampled by human beings. They're susceptible to death, right? But research over the years have actually shown that more often than not, very predominantly, bacteria actually exist as a community, and this community is known as biofilm. I'm going to talk about what biofilms are as I go along. And so here on the right side of my slide is an image of a bacteria that I study in the lab. The name of the bacteria is Pseudomonas aeruginosa. So this bacteria is a notorious bacteria for chronic infections. Some of the chronic infections that have been associated with this bacteria includes like wound infections, bone infections, especially in diabetic patients, as well as lung infections in people who have lung genetic issues. I'm going to talk about them even as we go further down in this presentation. But what I actually want you to focus on right here is that this is a microscopic image of a bacteria that was of the surface of a granite pebble. Can you imagine how small a granite pebble is? And this is an image of what is on the surface of a granite. Those little stones that bricklayers used to make houses, or to build houses in Nigeria or even anywhere else. Imagine having this mesh of individual cells interconnected together and forming like a huge mass, if you can follow my laser pointer on the slide. And so research over the years have actually shown that bacteria, as opposed to my initial assumption that they exist as this single cellular free-floating cell, they actually exist as a huge community. And this actually allows them to be more dominant, to be more resistant to antimicrobial treatments, to be more resistant to harsh environmental conditions. So like I mentioned, these communities are known as biofilm. So the question is, what is a biofilm? So a biofilm, from the name, bio and film. Bio signifies life, right? From microbiology. Bio refers to like film is anything that is slimy, anything that is like slimy or like that is glossy, something that is sort of like slippery. That's what you refer to as a film, right? So biofilm, so it's sort of like something that has life and is slimy. So I'm just using that as a primary mode of like defining what biofilm are or what a biofilm is. So a biofilm essentially is a complex community of microorganisms. So if you want to be very specific, you can call it bacteria biofilm. There is bacteria biofilm, there is fungi biofilm, but viruses, as we know, cannot exist outside of the host. And so you don't refer to them as a biofilm because they're not living outside of the host. And so we typically don't have viral biofilm. So a biofilm really is a community of microorganisms that are attached to one another. So this microorganism community could include different species of bacteria. So you can have Salmonella in there, you can have Bacillus in there, you can have Pseudomonas in that community. You could also even have fungi as well in that community. And this community is covered up in a polysaccharide slime or a polysaccharide matrix, and hence, the name biofilm. A very good example of a biofilm is the plaque that forms on your teeth when you do not brush for 2-3 days. Over time, I am assuming that my audience have like a very, a good amount of, a very basic background of biology. And we know that we have bacteria in our bodies. We have bacteria in our mouth. And if you don't brush in like 2-3 days, they're going to grow and they're going to multiply. And over time, they're going to form like a slime on your teeth. And that is a very good example of a biofilm. That's a bacteria community in your mouth. And so I'm going to move on to my next slide. And so the question now is, how do bacteria, individual cells, form a biofilm? How do they form a community? Again, biofilm is the same as bacteria community. If I call it bacteria biofilm, then I'm referring to a bacteria community, which is what my talk is on today. So the very first step in the biofilm formation process is attachment. So this is the step right here. If you looking at my slide, this is a step right here where individual bacteria cell would bind or attach to a surface. And the surface could be living surface or they could be non-living surfaces. So surfaces like, for example, people who have like diabetic patients who have like chronic wounds, the surface for biofilm attachment could be, on the open skin, the skin that is exposed. That's the surface. A single cellular organism or a bacteria can bind to that surface and begin to grow. And so the first step is attachment. Again, surface could be anywhere. Bacteria is found everywhere. So a surface could even be the surface of your, or the inside of your tap water at home. If you've noticed, sometimes over time, the pipes that, connect our, that is connected to our taps, over time becomes, you see like a clog. That's another example of the bacteria biofilm. So, or a biofilm, not necessarily just bacteria. And so the first step is the attachment step, where individual cells would attach to a surface. So the surface could be living surface or non-living surface. Then the next step, and this initial step is very reversible. So the bacteria could attach and detach, then they attach again, and then they detach. But over time, as the bacteria, divides and they increase and they proliferate, the attachment becomes more permanent. And so we move on to the next stage, which is the irreversible attachment, where the cells now adhere to the surface or even to one another. They can actually, if you remember the image I showed you earlier in the previous slide, the cells could actually attach to one another and not necessarily to a surface. And so they begin to multiply and they begin to grow. Individual cells will proliferate into more cells, and the biomass will begin to increase. Biomass, what I'm referring to as a biomass, is life mass, size. So the size of the bacteria increases. the quantity of the bacteria increases, the bacteria would multiply, and the cell or the biomass increases. And then you have the step known as proliferation, where the cell is each of the individual cells that make up the biofilm. The growing biofilm would actively divide, and they form this sort of like a 3D. I'm calling this 3D because there's height, there's length, and there's breadth. And so they form this sort of like a mushroom-like structure, right? And they go on to mature. So all these individual cells would, grow old, and you're going to get to the step where they're about to die. Some of them are going to, of course, die. But in the maturation step of the biofilm formation process, you would have like a nutrient gradient where cells at the peripheral, cells at the outer part of the biofilm have a better access to nutrients as opposed to cells that are inside the biofilm or at the bottom of the biofilm. And this is actually because of proximity. You typically have like maybe air, oxygen on the outside, you have like access to water, access to, other nutrients around the environment where the biofilm is being formed. And so cells that are on the outside of the biofilm have easier access because of the proximity, right? And over time, as this happens, the bacteria that are inside the biofilm would become dormant, right? And this actually has consequences for antimicrobial treatments. So the biofilm, the bacteria inside the cell will become very dormant, and they would just be there, not metabolically functional, and they're just there. And over time, the active cells, or the ones that are still functioning, would induce dispersal. So dispersal or dispersion is the process where cells that are inside the biofilm, the mature biofilm, actively leave the biofilm. They leave the center of the biofilm, just like you can see in the upper right corner of the slide. And so dispersed cells will then go on and then start the process all over again in a different surface or in a different place. And so what we actually have come to realize over time, from research, we have actually found many people have actually, not many people, a few people have said that, when we see bacteria in nature existing as unicellular, single cellular multi-cells, it's because they're actually in the process of transferring, moving from one biofilm to go from another biofilm. All right, so that, I think at this point, everyone should know what bacteria communities are or what biofilm are. So a bacteria community is an example of a biofilm. A biofilm is a community of microorganisms. And so if I want to be specific, bacteria communities are literally, a mesh or a biofilm or a bacteria biofilm. So the next thing I'm going to be talking about right now is why should I care about bacteria biofilm? Why should I care about bacteria communities? Why should we all care about bacteria communities? The major reason why we should care about them is that biofilm is everywhere. Bacteria is everywhere. You can find bacteria in the coldest of coldest places. You can find them in the hottest of hottest places. You can find them in lukewarm places, and places like, your homes or in your kitchen, everywhere. They're even on your body. Bacteria make up like a very huge amount of our body, of our system. And so they're everywhere. And that's why we should care about them, because they contribute as much as they're harmful to us. There are some of them that are also really beneficial to us as human beings. And so some of the places where you can find a bacteria, number one is in the wound infection. So here is an image of wound infection that's, and here is actually Pseudomonas, the bacteria that I work with in my graduate program. And here you can actually see the green-like color because the bacteria is greenish. It has the ability to produce green pigment. Here, so this is formed by bacteria biofilm. You can also see bacteria biofilm being formed in your water pipes. And that's why from time to time, it's very necessary that, our water tanks that we keep in our homes, we're meant to wash them out, after a while, because sometimes bacteria can clog, can settle and, form like this huge mass, and that could be really gross. And then also, like I mentioned, plaque on your teeth actually formed by bacteria uh, biofilm. They're actually formed by bacteria biofilm. If you notice, all those biofilm are actually slimy. You can see that they're actually slimy and glossy. They have like a shiny, glossy, slippery surface. And then lastly, this is actually very beautiful. It's a national park here in the US in Montana. It's the Yellowstone. And this beautiful mat that you actually see, it's a hot spring. And this beautiful mat that you actually see here is actually biofilm, bacteria biofilm. So a huge community microorganisms. So they're actually everywhere. They're everywhere. And so we can't choose to not care about them, because whether or not we care about them, they are part of us. So again, what are the benefits of bacteria existing as a community? The first and the most important benefit is that it allows for individual bacterial cells to communicate with one another and to collaborate with one another. And there is strength in synergy. When you have an individual cell, they're much more vulnerable to antimicrobial treatments. But when they're in that community that is now covered by an extra mesh of polysaccharide, it shields that community, it shields the bacterial cell that is inside that community from antimicrobial agents such as antibiotics or antiseptics. And so it allows bacteria to communicate. So bacteria actually do communicate, even though they don't communicate verbally. They communicate by producing certain molecules that helps the other bacteria to know that I'm here. They communicate. So it allows for bacteria to communicate. It allows for them to transfer antibiotic resistance gene from one bacterial cell to another. It allows them to also be resistant to antimicrobials. So I'm using antimicrobial resistance here because there are other antimicrobial substances that are not necessarily antibiotics. So antiseptic, reagents, for example, like your bleach is also antimicrobial agent. So when bacteria exist as a biofilm or as a community, they are much more resistant to antimicrobial treatments. And also they are much more resistant to a human being's or a host immune response when they exist as biofilm. All right, so now I'm going to be focusing on how does this community, what actually makes this community more resistant to antibiotics or antimicrobials generally. So the first factor that actually contributes to the resistance of bacterial community to antimicrobial, maybe I should probably just say this before I go on. So if you don't know, when a bacteria exists as a community, they are about 1,000 times. Well, I don't have the data here, but they're a ton of times more, I'm saying a ton, maybe that's too specific, but they're much more resistant to antimicrobial treatments than when they exist as single cellular, unicellular organisms. Okay, and so some of the factors that actually contribute to that is first, when they exist as biofilm, right? Like I mentioned, there's a nutrient gradient. And so you have cells in the center of the biofilm being dormant and less accessible to nutrients, whereas cells that are on the outer part or towards the surface of the biofilm are much more active and they're exposed to, they have a higher access to nutrients. And what that actually does is that you remember that if some of you have background in microbiology, some of the mode of action of some of the antibiotics that we use is that some of these antibiotics actually target cellular processes in active cells. And so if you apply an antibiotic to this biofilm, not only does the outer slime, the slime that actually covers the biofilm, the outer slime, acts as a covering, and somehow it would reduce the penetration of antibiotics into this biofilm. Also, because only bacteria on the outer part are active. Those ones might die, but then the ones in the middle that are dormant would not have any, they're not going to be killed because they're dormant anyway. And so because the antibiotics that we use, many of them actually target cellular processes in active cells. They would only be able to effectively kill or destroy cells that are active. So the other factor that also contributes to antibiotic resistance is the oxygen gradient. So you have cells on the outer part, more exposed to oxygen in comparison to the ones that are in the middle. And so if you go from the outer part of your biofilm into the biofilm, there's a reduction in oxygen concentration. And that also, of course, contributes to the activity of the biofilm. You have less active biofilm in the center, and so they are less susceptible to killing by antimicrobial agents. The other thing that I mentioned already is that there is a difference in antimicrobial concentration. Again, because you have the slimy surface that covers the biofilm, it reduces the penetration of antibiotics into the cell. And so most of the time, only bacteria that is on the outer portion dies. And then the ones in the middle, they don't even have access to the antibiotic, that has been applied to the biofilm. And so over time, they would survive. The ones in the center would survive. And the ones on the outer part of the biofilm would probably die. But then the ones in the center will survive, and then they're going to proliferate, they're going to divide again, and then they'll form this huge mass again that never goes away. And then lastly, the individual cells that make up the biofilm actually communicate with one another. And there is something that we refer to as quorum sensing. I'm not going to go into the detail of that, but it has something to do with cell-to-cell communication between one bacterial cell to another. And when the bacterial cell within the biofilm communicate one another, they sort of induce or tell each other, well, it's time for us to produce efflux pumps that would, contribute in pumping out whatever antimicrobial that's being, administered to the biofilm. And so when you're administering antimicrobial agents to the biofilm, these pumps would be actively pumping them out of their cell. And so the bacteria community never goes away because they're working together in unity. All right, so what is the implication of this? So the implication of this is that this bacterial community have been associated with chronic infections. So chronic infections are infections that never go away. So an example of a chronic infection is diabetes. Another example of chronic infection is what's it called now? I wouldn't consider cystic. I can't. Okay, so it's cystic fibrosis, and it's a genetic infection that is typically found in people here in Caucasians, in white people. It's just sort of, how sickle cell anemia is more predominantly found in Black population. Cystic fibrosis is a genetic disorder that's typically found within, the Western population. And so my bacteria that I work with in my graduate school research, Pseudomonas aeruginosa, has been attributed. This is just one bacteria. And of course, we know that there are tons and tons of bacteria. There are different species of bacteria. And so here is a graph that I got from the CDC website. On the y-axis of this graph, you have the estimated cases of multi-drug resistant Pseudomonas aeruginosa in hospitalized patients. And then on the x-axis, you have the different years. And if you notice, in 2012 alone, about 46,000 people had multi-drug resistant Pseudomonas aeruginosa infection. So they were not resistant to just one antibiotic. They were resistant to multiple antibiotics. So that's what multi-drug means. And In the most recent year, which was 2017, about 32,600 people had multi-drug resistant Pseudomonas aeruginosa infection, which is kind of interesting because this is just the US. If you're considering other bacteria, this is definitely going to be more. And now if you consider this in the context of North America, and then you go on to Asia, and then you go on to Africa, then I'm sure that this data is going to be even much more. So again, this is just to show you the impact of bacterial community and how this is contributing to antibiotics or antimicrobial resistance. All right, so I'm going to move on to the next thing that I want to talk about. And the thing I'm going to be talking about next is what are the potential impacts of bacterial community. I previously just spoke about why I should care about bacterial communities. Why I typically should care about them is because they are everywhere and they cause chronic infections, and they are multi-drug resistant, or they're resistant to certain antimicrobials. So now I'm going to be talking about the impacts of bacterial communities. So I know that many times when we hear about bacteria, we always think they're bad. But I'm also telling you there are good bacteria. Bacteria isn't necessarily always bad. Some of them are actually good and they're useful. We use them. So some of the impacts of the first one I have on here is that they are used in sewage treatment plants. So I don't know, I know that in the University of Ibadan, there's one huge sewage treatment plant towards Greek faculty. I don't know, I don't remember, but there's one huge sewage treatment plant in the university, and in one of the compartments, there's actually a mass, like a mesh of bacteria that they use to treat the waste. So you have your waste from all of the halls of residence, and from all the different faculties in the university, and they channel them to this sewage treatment plant. And so here, what you see here is just like a picture of it, and then this is just sort of like a schematic diagram or description of what actually happens in this setup. So you have your influent, which is your wastewater, the raw wastewater that's coming directly from some of the buildings in that community. And then it goes through a biofilter that has biofilm. Like I mentioned again, biofilm is literally just community of microorganisms. So you have the biofilter that has, bacteria, like a community of bacteria and what these bacteria actually do is that they some of them are bacteria that can break down nitrate to nitrogen. And then the nitrogen is then, released into the atmosphere. And so what they actually do is that they break down certain molecules in the waste that is being, put into this big, huge pond or container. And some of this bacteria also sort of detoxify some of the chemicals that you have in your sewage plant. Many things go into sewage. Sometimes bleach, the amount of carbon associated molecules in some of the waste that we get rid of in our homes is really high. And some of these bacteria use carbon as their source of, as part of their nutritional cycle. And so in the process of them using it for their own benefit, they're also like breaking down those huge, chemicals that you would typically find in your wastewater. And so bacterial biofilm doesn't necessarily have to be, bad. Sometimes they're actually beneficial. And also you probably have heard of, I'm going to go into it later on, you've probably heard of probiotics, and I'm going to go into all of that later on. So we also have, interestingly, we know that bacteria form a very huge part of human beings. We have bacteria in our skin. We have them everywhere in our body. We have them in our nose. We have them in our mouth. We have them in our ears. We have them, everywhere on our skin, everywhere, even in our gut. Your gut is your gastrointestinal tract, right? So your intestine, your large and your small intestine. So we have bacteria there as well. And I'm just going to go over this process that you can see on the slide here. So you have a microbiota. So what microbiota actually means is micro, small, biota, life, bio. Yeah, so it just means the bacteria that is in your gut, the microbiota in your gut, so the bacteria in your gut. So recent research has actually shown that the bacteria in our gut actually are there as biofilm, like as they're there as a community rather than individual cells. And so what actually happens is that in your gut, you have your epithelial cell that lines your intestine, the internal walls of your intestine, and you also have a mucous layer that actually allows food or debris, everything to just flow through easily through your gut. So what actually happens is that this bacteria biofilm some of them would disperse, and they form this individual cells that are now known as pantobiotes. And what this actually means is panto, when you hear panto, it means something that is bad, pathogenic, meaning it's something that causes infection. So a pantobiot is, in this case, is a bacteria that causes infection or produces toxin that is bad for the host or for the human being that has it. So some of the bacteria would leave the biofilm, form individual pantobiotes, which are harmful, and then they are going to cross the mucous layer. The mucous layer on the lining of your intestinal wall actually allows for stuff to flow through easily, and it sort of acts like a barrier in between to prevent bacteria from penetrating into your epithelial cell. So they cross the mucous layer, and then they penetrate into the epithelial cell, and then they produce toxins that would eventually cause things like running stomach, diarrhea, dysentery, and whatnot. And then when you go to the doctor's office, they would administer antibiotics for you. And so, of course, another impact of bacterial biofilm is that they cause infection in our stomach, even in our gut. Right. So another interesting impact of biofilm is that they cause chronic infections. I already mentioned this before. So here is an image of a chronic infection that is caused by my favorite bacteria, Pseudomonas aeruginosa. And this is actually caused when you use a contaminated contact lens. And so for people who are using contact lens, make sure that you're keeping it properly and you sterilize it before you use it each time. Because if there's a bacteria somewhere and you stick it on your eyes, because contact lens sort of reduces, the production of the fluid from your eye, it can sort of, induce or encourage the growth of bacterial biofilm. So here is keratitis, it is an infection of the eye caused by biofilm. And it's worse when it is caused by biofilm because the infection would just never go away. Yeah. Also, the bacteria Pseudomonas aeruginosa forms biofilms on the lungs of cystic fibrosis patients. So we refer to them here as CF. So you know how sickle cell is sort of rampant in Nigeria? We also have CF, not as rampant in Nigeria, but in CF there are CF patients, people who have CF, cystic fibrosis. You can look it up. And these people are actually born with it. Their life expectancy is I think the highest life expectancy for CF patients should be maybe 40. I don't have the data, but they don't live as long as normal human beings. And a lot of research is going on to try to figure out how to, help them live longer, by, combating, bacteria, because they never, so the problem with CF is that due to the mutation, the genetic mutation, they produce very sticky mucus. They don't produce as much water in their the epithelial cell that lines their lungs. And again, the mucus in our lungs, the ones in our gut, they're necessary because they help you to push out pathogens so that they don't stay there and continue to grow. So when you have a sticky mucus and you have something that is just, you don't make that much water because of the mutation in your DNA. When a person inhales dust that has bacteria, or inhales hair that has bacteria in it, it just stays in the lungs, in the airway of that person for a CF patient. And for especially one of the predominant bacteria that's been associated with CF patients is Pseudomonas. And here is a lung. This is a lung tissue of somebody who had CF. And here you can actually see the greenish sediments of biofilm. And last chronic infection I have here is green nail syndrome, is a bacterial infection of your nails that just somewhat never goes away, and it's caused by Pseudomonas aeruginosa. Yeah, this is a photo on my part. All right, so the potential impacts of bacterial communities have been spoken about. So now we're going to move on to how we can actually explore or exploit these communities for public health benefits. All right, so the very first thing that, again, I've been able to share that there's strength in community. I'm running out of time. There's strength in community. And so the first thing you want to do, if you want to try to, if you have a huge army of soldiers, one of the ways that you can actually penetrate their camp is to isolate each individual soldier and try to fight individually with them. So you can bring that strategy to bacterial communities as well. There's vulnerability when the person is isolated from their community. And so what we try to do, one of the strategies that we're doing or that is being explored in biofilm research, is that we try to induce dispersion. So we know that bacteria predominantly exist as biofilm. So if we try to cause the biofilm to disperse so that you have individual cells, then you can apply antibiotics to those individual cells that are now much more susceptible to treatment. So some of the strategies that is being used is, dispersal strategies. You try to introduce, for example, enzyme degradative enzymes that would break, the covering, whatever mesh that covers the biofilm. I don't want to use the actual terms, that's why I'm using those. Mesh that covers the biofilm so that, that allows for the bacterial cells to get dispersed from those community, and then you can apply antibiotics to that dispersed biofilm. Another method that is being used because some people have issues with their gut, and they just never well. We're just thankful that we have a very healthy gut. Another strategy that's been used is the use of probiotics. And I don't know if you guys are familiar with what probiotics is. Probiotics are, bacteria that are good for us and they help in our digestion. They also produce toxins that are harmful to bad bacteria. And so many times people will say, oh, take yogurt. The reason why they ask you to take yogurt is because there are good bacteria, lactobacillus species that you would typically find in yogurt. And so that's why they say you should take it, because it's good for your gut and helps your health, your gut. It improves the health of your gut. Okay, so what are the strategies that probiotic uses to disrupt or to disrupt biofilm? The first thing is that the probiotic bacteria, whatever bacteria, most of the time they usually belong to the Lactobacillus genus. I'm sorry if you don't, for people who might not know what that is, but yeah, that's the name. You don't need to know all those names, but yeah. Probiotic bacteria would compete for attachment with the harmful bacteria, and that would sort of reduce the ability for harmful bacteria to establish itself and form a biofilm. The other thing that probiotics do is that they produce toxin, and these toxins are known as bacteriocin. And these toxins are harmful to bad bacteria, and so they're going to kill or sort of inhibit this bacteria from forming a biofilm. Another thing that they also do is that they produce biosurfactants, and so biosurfactants are sort of like they're fluid in nature, and they allow. I think that they're fluid in nature, and I think that what they actually do is that they enhance adhesion to the surface, and so they reduce adhesion to the surface. And so when you have biosurfactants being produced by probiotic bacteria, it reduces the attachment of harmful bacteria to the walls or to the epithelial cell that is inside the guts of a person. Another thing is that they also produce certain molecule. I don't want to mention what these are, but they produce certain molecules that sort of activates the immune system of the host. And so when the immune system of the host is activated, they can be recruited to the site where the biofilm is being formed, and they can sort of, stop the biofilm from progressing, the biofilm formation from progressing. And so just to kind of conclude my presentation. So today I've been able to talk about why community is necessary, that there is so much strength in a community. Imagine just little bacteria coming together and, being a problem to human beings, giant human beings like ourselves. And so there's strength in community. And so I'm just encouraging everybody to make use of the community that you have. The community that you have at your homes, your parents, your siblings, the ones that you have in school, your friends, the ones you have in your different religious gatherings that you might have, just make sure that you make use of your community. And also that bacterial communities are a lot more stronger when they exist. Bacteria are a lot more stronger when they exist as a community, as opposed to when they exist as individual cells. And lastly, just like we see that bacteria are actually useful. We have good bacteria like probiotics, and we also, see that they're useful in our sewage treatment plants, that not all bacterial communities are harmful. So with that, I am going to be taking any questions that anybody might have. Thank you.
Thank you very much for the wonderful session. It's been an interesting one. I think I'll be taking lots of probiotics now, lots and lots of them, so that whatever form of bacteria we might have, or biofilms of bacteria, we'll ensure that we have the good ones present. Thank you for the beautiful lecture. I know we are
Hello, good evening, everyone. Can everybody hear me?
Yes, we can.
Yes, I can.
Yes, we can hear you.
So I'm just going to step in for Idris. I think he got logged off due to his network connection. So we're in the question and answer session. So if you have any questions, if you want to speak by yourself, please just raise your hand. Then we'll give you the permission to unmute yourself. Or you can drop your questions in the chat box. I believe we have a question, Victoria. So it's from Martin. It's from Martin. So his question is, how do you differentiate between prebiotics from probiotics? Did you get the question? Hello, Ms. Victoria.
Yes, sorry. I'm actually juggling between my computer and my phone. Sorry about that.
No problem.
I got the
Did you get
question.
the question?
Yes. So what's the difference between prebiotics and probiotics? So I know, so probiotics, like the name suggests, probiotics, life biotics. So probiotics is a food, for example, that contains live bacteria. For example, yogurt. Yogurt is a very good example of a probiotic. Pap or ghee, the very fermented pap is also a very good source of probiotic as well, because they have good, microorganisms, for example, like yeast that you would typically find in ogi that we take in Nigeria. So probiotic is a food item that contains live bacteria and helps your gut. It helps to helps in digestion, produces toxins that are harmful to harmful bacteria, and generally just helps. I don't want to go into all the details, but prebiotics, I was just checking Google right now because I'm not really too familiar with prebiotics, but according to what I just saw, it said that prebiotics are fiber, food that are rich in fiber that helps, sort of like feeds good bacteria in your gut, according to what I saw on Google. So I am not really vast in that area, in the area of prebiotics, but I do know that probiotics are food items that have bacteria in them. But according to what I saw on Google, I saw that prebiotics are actually, food items that are good for good bacteria. So I guess, I don't know if that answered your question.
Hello, Martin. Does that answer your question? And thank you, Ms. Victoria, for that answer.
You're welcome.
Okay. So there's another question from Tolu or Modular. So it says, do we have a threshold of bacteria that could either benefit or harm our gut health?
So I think it depends on what you're referring to as threshold. So a bacteria that is bad, regardless of how much of that bacteria you have in your system. A bacteria that is good, regardless of how much of that bacteria that you have in your system. So are you referring to bad bacteria, or you're referring to like any type of bacteria? I also think that I should mention that sometimes you can have a bacteria could be harmful or might not be harmful depending on how good of a human immune system that you have. So I'm going to use Staph aureus for an example. Staphylococcus aureus. So Staph aureus is a bacteria that is typically found on your skin. I probably have it on my skin right now, and I would want to think that many of us have it on our skin. But if you notice, sometimes when kids are growing up, they have boil, and it's one of the major causes of boil, BOIL, that children usually have, and that usually happens when Staph aureus is a opportunistic pathogen. It actually is not as harmful when you have a good immune system, when your immune system is up to par, but when you become immunocompromised, it can switch. It can take advantage of the fact that you're immunocompromised and trigger an infection. So I don't know if that answers your question. So I'm asking this question as to to bad bacteria, or are you referring to opportunistic bacteria? So yeah, I think that would help me to better answer your question. All right, so all of the above. All right, so if you have good bacteria in your gut, if you have probiotic bacteria in your gut, of course, that would mean that you would have a very healthy gut. If you have a little of it, you would have a good health. You have a good gut. If you have much more of it, well, the better, and the even the greater for you, right? And so, because this good bacteria is going to suppress harmful bacteria that you might have in your gut as well. If you have a bad bacteria, then that's very, very bad. Some people really literally have to do like a gut transplant where it's a very bad issue for people that have, when you have bad bacteria in your gut that produce toxins that will sort of eat up your epithelial cell, cause like a lesion in your gut, in the internal walls of your gut. And that could sometimes, project into ulcer. And I think very recently, maybe not, there's beginning to be like an interrelation between your gut microbiome, like the health of your gut, and stomach cancer. So that is, yeah. So you don't want bad bacteria. You do not want bad bacteria, and we talk less of having much of that in your guts. And then when you have bacteria that are sort of like in the middle, they're not bad when your immune system is good, but when your immune system is bad, they become really terrible. You don't want to have too much of that in your body as well, because our immune system sometimes can go weak when we fall, when we medications, and because some of the medications that we take could suppress our immune system. So you don't want to have too much of, we call them commensal bacteria. You don't want to have too much of that as well. So I think a healthy human being sort of has like a good balance between, commensal bacteria, probiotic bacteria, and maybe just a little bit of harmful bacteria.
Thank you, Ms. Victoria. So does that answer your question? While we're waiting for Tolu's response, I believe Martin's hands are raised. So Martin, you can unmute yourself to ask your question. Hello, Martin, can you hear me?
Hello, good evening, Jonathan. There are some people that they use my own personal link and it's showing my name. So I would like
so whoever is the Martin that raised his or her hands up, you can unmute yourself to ask your question. Yeah, you just waved now, so you can unmute yourself to ask the question. Is there a way that I can?
Good evening, everyone. Good evening, everyone. Okay, thank you so much for this wonderful presentation. Okay, my name is Genesis Ulakom. Thank you for the wonderful presentation. Okay, my question is kind of situation I experienced during COVID, during lockdown we had. Now I discovered a lot of persons were taking, ginger, garlic, and all of those things to maintain their gut health, like to keep fit. That period, a lot of persons were going about it. Virtually everybody was going about with his or her nose masks to keep away from, people who are already infected and just to be healthy. So my question is, do you advise, or do you think taking probiotics or prebiotics, food that contain or that can enhance the growth of probiotics can suffice, or do you advise that people should start taking probiotics or prebiotic bacteria or PAP, as the case may be, or will PAP be able to do that kind of job of activity to keep fit with people who have a head gut problem, or having people who have head respiratory diseases, like their respiratory system is affected and looking out for alternative treatment measures. So do you advise taking probiotics food as a measure to curtail or stop or reduce or treat that kind of sickness?
Okay, that's a very good question. So I think that question, I think that how I'm going to respond to that question, I'd say is that it depends on what is causing the infection that people have. So for example, COVID. COVID is a viral infection, right? It's not a bacterial infection. And so many people just assume, okay, if I have a viral infection, you can't use the same strategy to treat viral infections to treat bacterial infections, right? You can't use antibiotics for a viral infection. People have malaria. They're saying, oh, you should go get antibiotics. Antibiotics are for bacteria. They're not effective against protozoans like Plasmodium, which causes malaria, for example. So you shouldn't be taking antibiotics for a viral infection, such as COVID. So that said, I do know that when you have respiratory infections such as cold or catarrh or COVID, for example, people would say, oh yeah, you should actually take fluid. You should take a lot of fluid, and you should also take lemon, ginger, honey. And the reason why you're taking those things are not necessarily because they are probiotics. Of course, honey, ginger, they don't have bacteria or anything like that in there. I think it's more to increase the fluid in your system, especially for people who have respiratory infections. Respiratory infections, many of the times, sort of is accompanied with congestion in your nose. And so what you're trying to do really is to sort of decongest what is in your nose. And, taking ginger, garlic during the times when you have respiratory infections, such as COVID, is just sort of to increase the fluid that you have, in your system, and, sort of to declutter whatever thing you have going on in your olfactory system, in your nose. So I know that lemon and ginger probably has certain chemicals or certain biomolecules that sort of enhances immune response, but I don't know all the detail about that. But that's just in regard of sort of somebody, thank you, Nasir, antioxidants. That's in the context of viral infections. Now, if you're talking about bacterial infections, if a person has issues with their gut, the first thing the person wants to probably figure out is what exactly is wrong with my gut? Am I having diarrhea because I keep taking infected, improperly cooked food? Or is it because there's an imbalance? Because usually when people have issues with their guts, it's because they have more bad bacteria in their gut, or more commensal bacteria in their gut and less probiotic bacteria in their gut. And when there's that imbalance, it causes a lot of issues for people. So if that's the case, maybe having them take more probiotics could help. But if it's much more than bacteria, if it's something else, maybe another strategy, maybe taking probiotics might not necessarily fix that problem. So I think it depends on what the problem actually is before you can determine what solution to prefer in different situations. I don't know if that answered your question.
Thank you, Ms. Victoria. Genesis, does that answer your question? Okay, yes, that answers this question. Thank you. So there's a question in the chat box from Marina Dubar. I'm sorry if I didn't pronounce your name properly. So it says, is the pressure sore in bedridden patients caused by bacteria? Did you get the question?
Yes, I got it. So I think it depends. You cannot say that. So sores, wounds, burns, these are, I wouldn't call them infection. You don't call it an infection until they're actually infected by a bacteria. So if for example, I'm going to give an example where you're running on the road and you sleep and you fall, right? You're going to have a wound, right? That wound is not caused by a bacteria, right? And so what actually happens is because you have bacteria on your skin, right? The bacteria, or maybe even in the environment, if you're not taking care of the sore properly, right? Bacteria can get into that sore. Bad bacteria can get in there. Commensal bacteria can get into that sore, and they can begin to establish themselves and, form a biofilm that is very difficult to eradicate. And they can even make the sore not heal because the antibiotic just wouldn't work. Or you probably need to take like multiple antibiotics to, have it work against, the biofilm. And so the sore in itself is not necessarily caused by bacteria. But when a sore is present, if a person is, bedridden on the bed for a while, because of the pressure, staying on one side of the bed, it can induce sore, or it can induce wound, if I would say that. And then if the bacteria gets into that wound or that open skin, then it can establish itself and even make the infection, the sore, even worse. I don't know if that answered your question.
Thank you. Does that answer your question? Then there's another question. Okay, you're welcome, Mina. There's another question from Nasty Rubin. So it says, I'm particularly interested, in the chart showing decrease in Pseudomonas aeruginosa resistance from 42,000 plus to 36,000 amongst US citizens. So the first question is, does that mean they are strictly adhering to proper usage of antibiotics? And two, are the data presented a follow-up one from the same patient or a different patient?
Oh, those are actually really great questions. So I'm gonna go back to the chat so everyone else understands what. Okay, yeah. So this is the chart right here. Yeah, there's a decline, right? I'm not sure how this data was collected. I just went on the website. I don't know how the data was collected. It could be that maybe you have the same patients in the case study that were they were assessed in 2012 that were also assessed in 2017. I'm not sure. I don't know. But what I would actually say is that maybe this data was gotten from, just hospitalization. So people, of course, if you go to the hospital, in a country like this where data collection is actually very important. For example, if you have COVID, you have to report to the county that you have COVID so that they can take you down, like, oh, you had COVID, and then they'll take some of your information as well. So I don't know if this is how this data was gotten. I don't know if there's a sort of like a follow-up, to try to assess whether individuals who were assessed in 2012 were also assessed in 2017. I have no idea. But I think that my biggest guess would be that they just sort of, counted the number of people that had Pseudomonas aeruginosa every year, regardless of whether or not this infection is reoccurring or is persisting over this period of six years. I don't know. So that question, I wish I had the answer, but I do not know. But that's a really great question.
Okay. Thank you very much. There's a follow-up question from Tolu. So the question is, how do we know which kind of bacteria we have in our system?
All right, that's very simple. Inside our system or outside? Okay, so the literature actually has published, I think it should be in the literature or in publications, of types of bacteria that you can typically find in different parts of your body. Type of bacteria that you can find in your reproductive system, type of bacteria you can find in your skin, on your skin. Staph aureus is one of them. type of bacteria that you can find in your nursery, in your hives, whatever. So I would think that there will be a publication that would have that would sort of address, would, you know, tell you what type of bacteria you can find in different parts of your body if you're a normal human being. I mean normal by normal, I mean that you're not sick. That's what I mean by normal human being. But the easiest way to do that, if you want to go the experimental way, would be to like take swabs of different part of your body and then culture them in the laboratory and see if anything grows. And if nothing grows, it doesn't mean that the bacteria isn't there. It could mean that you're not using the proper bacteria. I said bacteria. You're not using the proper growth media to grow the bacteria. So there are two major ways you can do it. First, you can go into the literature, you know, go on Google or go onto Google Scholar and just search, you know, what are the types of bacteria that you can typically find in human beings or in different parts. Yeah, and I'm sure that a few things will pop up. And then if you want to do it for yourself, because that is like based on, you know, research on other people. If you want to do it for yourself, the easier way would be to just take swabs of different parts of your body and then just culture it in a laboratory so you can know what type of bacteria is in your different system. But I do know that, you know, lactobacillus is typically found in your gut because they're necessary. Because number one, because of the pH in your gut is very acidic, and you can only find bacteria that, you know, has tolerance for acidic environment in there. The reason why you can also find Staph aureus on your skin, Staphylococcus aureus on your skin, is because, you know, your skin is the salinity. Our skin is a little bit salty. Yeah, if you taste your sweat, it's a little bit salty. That's gross though, but don't do that. So yeah, so that's why Staph aureus can survive in a fairly salty environment. So that's why you can find it in your skin. So yeah, different bacteria in different places. Even in your mouth. One of the, I can't remember from the top of my head, but there are different bacteria in different places in your body, of course. And I think you should be able to find it online. So yeah.
Okay, thank you. Adeleke Dorcas has a hand raised up. You can unmute yourself, Dorcas. Hello, Dorcas. Okay.
Hi, good evening, everyone. My name is Adeleke Dorcas. My question is, you said we have some bacteria that are good for us and all that. In a case whereby, someone is sick and a different antibiotics is being prescribed for the person to use, and it maybe eradicate or kills the good bacteria that's meant to keep the person safe. Mm-hmm. How does the bacteria comes back? Does the bad one replicate them, or do they come back again? I just want to understand that.
Oh, that's a very good question. Thank you for that. So that's why, in addition to not, drug abuse, it's drug abuse to take a medication that has not been administered to you, especially antibiotics. Some of the problems that we are facing right now in antibiotic resistance is due to misuse of antibiotics, right? The thing that people don't know is when you take antibiotics, the antibiotics is not just going to kill, the bad bacteria. It's also going to kill the good bacteria too. That's something that people don't know. And so we should be very careful when we're taking antibiotics. I don't know, there's some people who, after they finish taking antibiotics, especially for women, they produce or they have like yeast from their reproductive system or from their reproductive system after they finish using antibiotics. Is because, the microbiota in their reproductive system, the good one is already depleted. And so the ones that are commensal would begin to manifest or would begin to even grow more, thrive more, because you don't have the good ones checkmating the bad ones. If that makes sense to people? So when we take antibiotics, you're actually killing both the good and the bad antibiotic. And that's why taking antibiotics should be modulated. It should be yeah, it should be modulated. It shouldn't just be you just taking it and taking it and taking it. It should be something that should be modulated, because you also want to give your body enough time to recover from that depletion. And after you finish taking antibiotics, of course, you don't take antibiotics for like an entire year. You probably take it for like a few days in a year, maybe, if a person is very healthy. And so after a while, the bacteria that is left, the good bacteria that is left, would divide again, and then it's going to populate, become more, and then they begin to be more effective in checkmating the bad bacteria. I don't know if that answered your question.
Okay, thank you.
Yes. Okay.
Is your question answered? Is there any follow-up?
Yeah.
Okay, no. The next question on the chat box is: do all bacteria from biofilm? Do all bacteria form biofilm? Do all bacteria form biofilm? That's the first one.
Okay, yeah. So I try to stay away from absolute statements because I don't want to generalize. I wouldn't say that all bacteria forms biofilm, because I don't have data to prove that, right? I'm a researcher. When I'm making statements, I don't make like holistic statements like that. But I would say that many bacteria form biofilm. And from research, we've seen that the predominant mode of growth of bacteria in nature is biofilm. So I would want to think that many, if not most, bacteria actually forms biofilm, but I can't tell if all of them form biofilm because I don't have the data to support that.
Okay, the next one goes like this. One of your
I can't hear you anymore if you're speaking.
illustrations suggested that new biofilms come from existing biofilms to themselves or to the surface to form a. I don't know if you get.
You're
sorry, let me just say the question. It is permit me to say the question. So the next question is: one of your illustrations suggested new biofilms come from existing biofilms.
Like,
what? Sorry, Idris.
Can you hear me?
Yes,
yeah, you're breaking. Let me take the question. Okay, so like as I was saying, one of your illustrations suggested new biofilms come from existing biofilms. What determines when the bacteria can adhere to themselves or to a surface to form a biofilm, and when they can detach and become free again to begin a new cycle? Did you get the question?
Yes. So the question is asking what actually makes the bacteria to determine whether or not to attach or whether or not to leave the biofilm. So yeah. So there's a whole. I forgot to add this disclaimer to my presentation. The presentation I gave is very vague. It's sort of like just on the surface. There's a whole lot to biofilm information. There's a lot of stuff going on that I did not mention. So this is just a very primary, elementary presentation that I just gave. So what actually makes a bacteria to adhere? So there are a lot of factors. So number one, if a bacteria has flagella, so flagella is the little tail, or not all bacteria has flagella, but many of them have flagella, and that actually allows the bacteria to swim to the surface where they're going to attach, and they're going to attach if it's a good place for them to attach. If they feel like, well, I think I'm going to get nutrients here, they're going to attach there. So the surface could be the surface of a person's teeth, for example, or the surface of it's most likely going to be a place where they have access to nutrients. A place that is polite. Nobody will want to go to a place where they're going to be poor, right? Human beings, yeah. So I think that bacteria is also wise in that regard as well. So if they have anywhere that is, good for them to adhere, places where they're going to be accessible to nutrients, they're going to want to attach. And some of the things that they use to attach is their flagella, and they also use, it's a shorter appendage, it's a shorter, what's it called now? organelle in a bacterial cell. It's called pili. They also use that to attach to a surface. And then when they attach to those surfaces, it's not just one bacteria, multiple different bacteria would attach to that same surface. And then the ones that are attached will then begin to multiply, right? So a bacterial biofilm does not consist of, might not consist of just one bacterial species. It could be multiple bacterial species. It could be Salmonella, E. coli, different types of bacteria all meshed into one wholesome community. And then when the biofilm is mature, right, and there is uneven access to nutrients, the individual bacteria inside the biofilm begin to produce different chemicals. I don't want to go into like the nitty-gritty of it. They begin to produce different chemicals that sort of induces other bacteria to know that, well, it's time for us to leave again. We don't have as much access to nutrients as we did initially. So it looks like we're beginning to be congested, right? We need to get out. And so they induce degradative enzyme. They produce degradative enzymes that will sort of like break the outer covering of the biofilm, and that would allow for bacteria to leave the biofilm to go start up somewhere else. So, and again, they can swim to a nearby surface, they can swim to even a farther surface, depending on their whatever cues they get from the environment. So it's not a one size fits all type situation. It's dependent on whether the environment where they're going to grow is actually conducive for them to stay, and whether or not the nutrients that they have in that mature biofilm is enough for them. Once they realize that, well, we're getting congested and we think this is not good enough environment for us, they disperse and then they go on to colonize a new environment. So I don't know if that answered your question. Thank you for having.
Sorry, I was okay. Yes. Does that answer your question, Will?
Yes, I'm clear. Thank you.
Yes. Thank you. You're welcome. Does anybody have any other question? I believe there is none. Okay, so thank you, Victoria, for that wonderful question. It was very enlightening. I believe we all learned a lot from today's session. Me in particular, I also had the impression that bacteria existed as cellular organisms, and today it has been an eye-opener for me. So before we close, I'll call on Will to give the vote of thanks.
Thank you, Chinaza. Okay, thank you, Victoria, for coming to Science Cafe. Thank you for taking time out of your schedules to honor our invitation. And we appreciate the time you spent with us. And we've learned a lot, of course. And so we wish you the best in your graduate program and, of course, all of your future endeavors. Thank you very much.
Thank you so much. Thank you for having me. I'm so honored to be here. And thank you for being a very interactive audience as well. God bless you all.
Thank you, Victoria. So with that, we'd like to say thank you for gracing us. Thank you for gracing us with your presence. Thank you for enlightening us. Thank you for teaching us. And also thank you to the participants for asking very thought-provoking questions and for making this session very enjoyable. It was very nice to have you all in the session. And with that, I will say good night, everyone. Good night.